The overall goal of this in vitro bioluminescence assay is to determine the effects of different environmental circadian disruptors and enhancers on the cellular circadian rhythm of mammary epithelial cells from mammary glands at normal or disease states.
This method can help answer key questions in the environmental health field, such as whether environmental factors, including chemical compounds, affect the cellular circadian rhythm.
So the main advantage of this assay is that it can be readily adapted to a variety of cell types, so we can screen for environmental modulators of circadian clocks in different organs.
Demonstrating the procedure with me is Brian Estrella, a graduate student in our lab.
The human PER2 promoter fragment is obtained from a customized vector that contains human PER2P at the site between SAC1 and HND3 in the multiple cloning region. To cut the human PER2P fragment out from the vector, mix the following in a DNA, RNA, and nucleotide-free microcentrifuge tube: 2.5 microliters of restriction enzyme buffer, 10 microliters of vector, 1 microliter of SAC1 restriction enzyme, 1 microliter of HND3 restriction enzyme, and ultra-pure water to 25 microliters. Incubate in a heating block at 37 degrees Celsius for 90 minutes. When the restriction digest is complete, run the whole volume of the reaction on a 0.7% agarose gel containing 0.0001% ethidium bromide to separate human PER2P from the vector. Cut a piece of agarose gel containing a fluorescence band at 941 base pairs, purify the